Hi everyone, my name is Jian Guan from Nanfang Hospital, Southern Medical University in Guangzhou, a city in southern China. Thank you for inviting me to share some of my past work here. The topic I'm sharing today is COMMD10 enhanced radiosensitivity by disrupting copper-ion balance in hepatocellular carcinoma. Copper is involved in many biological processes such as cell respiration, ion homeostasis, antioxidation defense, and neuropeptide processing. Copper is an essential nutrient which makes it both beneficial and toxic to the cell. It was reported last year that copper induces cell death by targeting liponated TCA cycle proteins. But the connection between copper and cancer is more defined, as regulated copper-dependent cell proliferation and is an example of broad range of metaplasia. Curecancer is linked to a diverse array of cellular processes, including mitochondrial respiration, autophagy, and protein quality control. The dual role of copper has been demonstrated in numerous oncology studies. That is, within a specific concentration range, copper may promote cell growth or play a deadly effect. Which way to go is still unclear. We try to find clue from the patients. An elevated copper level of tissue and serum has been reported in multiple cancer patients, including liver cancer, breast cancer, prostate cancer, cervical cancer, lung cancer, and gastric cancer. It has been reported that the level of serum copper was negatively associated with radiotherapy response in cancer patients, which indicated that serum copper was an effective monitoring index for the radiotherapy efficacy. In addition, recent clinical trials suggest that the copper channel could function as an adjuvant therapy to improve the effectiveness of chemoradiotherapy in multiple cancers. However, the potential mechanism remains unknown. Therefore, we take copper as the starting point to explore the new mechanism of radioresistance as the main place of copper metabolism. The liver is very important for maintaining copper homeostasis. Here we have 7 parts. Irradiation-resistant liver cancer cells were used to evaluate the relationship between copper and irradiation response, and their radioresistant characteristics were confirmed by radiation assay. The radiobiological parameters positively associated with radioresistance. The DREL SF2 and N value in resistant cancer cells were significantly higher than those in parental cells. In addition, a significantly higher level of copper was detected in resistant cells than parental cells. This suggested a potential role of increased copper in inducing radioresistance. In a microarray dataset with profile transcriptome in HEP2 cells, we find the increasing dose of copper was related to radiation response. Cell counting kit 8 showed that the liver cancer cells occurred toxic responses when exposed to copper over 300 micromole, while radioresistance could be found when exposed to copper lower than 200 micromole. In addition, EDU assay confirmed that the increased intracellular copper promoted proliferation regardless of radiation exposure. Furthermore, we find in vivo experiment after irradiation, the serum copper concentration, tumor volume, and tumor weight in the mice fed with copper were significantly increased than those without copper treatment. TEPA, a copper channel, significantly inhibited serum copper and enhanced the radiosensitivity of subcutaneous tumors in mice fed with copper. These findings confirmed that increased copper contributes to radioresistance, and TEPA was a copper-dependent selective radiosensitizer in liver cancer. Having established copper was a contributor to radioresistance, we try to find candidate genes responsible for the regulation of copper metabolism and radiosensitivity. Reportedly, common family decreased in 293 T cells treated with copper, which indicated that important roles in the regulation of copper metabolism. To verify which common protein member is involved in regulating radiosensitivity, HER2 cells were exposed to 0, 2, 4, 6, and 8 degree radiation, respectively, for 24 hours. It was shown that COMMD10 was the unique gene that was significantly decreased in an irradiation dosage-dependent manner in HER2 cells. To verify the potential role of COMMD10 in irradiation response, we constructed COMMD10 overexpressed and depleted liver cancer cells based on endogenous COMMD10 level. CCK8 assay and clonogenic assay showed COMMD10 overexpression significantly inhibited the proliferation of liver cancer cells, but COMMD10 depletion obviously enhanced the proliferation when exposed to irradiation. In other words, COMMD10 depletion induced radioresistance in liver cancer cells. It's very interesting that COMMD10 significantly decreased in the resistant liver cancer cells. It could be reversed when COMMD10 overexpressed, which is shown in Figure 2G and is supplementary to L and supplementary to M. We further validate the correlation between COMMD10 and radiosensitivity in clinical data from TCGA and GEO database. We found that COMMD10 low expression were predicted to be more radioresistant than COMMD high, which suggested that COMMD10 may be an effective predictor of radiosensitivity. To explore how COMMD10 increases the radiosensitivity of liver cancer, we conducted proteomics analysis. Gene ontology analysis discovered that proteins significantly enriched in regulation of cell death, which showed in Figure 3A. To further clarify their relationship, we treated COMMD10 overexpressed and depleted cells with several cell death inhibitors, such as apoptosis inhibitor, autophagy inhibitor, pyroptosis inhibitor, and the necrotosis inhibitor, the ferroptosis inhibitor. We found COMMD10 might sensitize liver cancer cells to radiation by inducing ferroptosis. We further studied whether COMMD10 modulated radiosensitivity in liver cancer cells through regulating ferroptosis. The level of lipid peroxidation at MDA were significantly elevated in COMMD10 overexpressed cells exposed to irradiation, and this could be significantly attenuated by ferroptosis inhibitor. Next, animal model was constructed to explore whether COMMD10 enhanced radiosensitivity through promoting ferroptosis. Tumors from COMMD10 overexpressed group were much smaller with higher level of lipid peroxidation than those in mock group. However, this effect could be reversed by treated with ferroptosis inhibitor. These results confirmed that COMMD10 enhanced radiosensitivity by promoting ferroptosis. To determine the underlying mechanism, we examined the genes after cells were irradiated. We found only the SLC7A11 decreased obviously in COMMD10 overexpressed cells and increased in COMMD10 depleted cells. Furthermore, COMMD10 depletion increased SLC7A11 expression, while COMMD10 overexpression inhibited its expression by Western blot. SLC7A11 plays an important role in glutathione synthesis. To confirm whether it's a downstream target of COMMD10 involved in the induction of ferroptosis, we constructed COMMD10 and SLC7A11 co-expression cells to detect the levels of glutathione. The results showed that COMMD10 overexpression decreased the level of glutathione compared to mock cells, and SLC7A11 expression attenuated COMMD10 induced inhibition of glutathione, suggesting that COMMD10 enhanced ferroptosis by suppressing SLC7A11 expression in liver cancers to irradiation. We further explored how COMMD10 inhibited SLC7A11 expression. The string database predicted two potentially interactive proteins of COMMD10, and KHG pathway analysis showed that these proteins were mainly associated with HIF1 signaling pathway and ubiquinone-mediated proteolysis. Thus, we verified whether COMMD10 interact with HIF1 alpha using immunoprecipitation, and the results showed that COMMD10 combined with HIF1 alpha in lomoxia and hypoxia condition. We further constructed COMMD10 N-terminal and COMMD10 C-terminal truncation mutants to determine the binding site of HIF1 alpha in COMMD10. This pull-down revealed that COMMD10 N-terminal could combine with HIF1 alpha. Moreover, in both irradiation and hypoxia conditions, the nuclear translocation of HIF1 alpha was promoted in COMMD10 depleted cells and was inhibited in COMMD10 overexpressed cells. The stain gives the same information. Interestingly, we found the binding site of HIF1 alpha in the promoter of SLC7A11. Overexpression of HIF1 alpha significantly increased the expression of SLC7A11 at both mRNA and protein levels. Furthermore, COMMD10 depletion could increase HIF1 alpha and SLC7A11 expression. Meanwhile, HIF1 alpha inhibitor YC1 could reverse the up-regulation of HIF1 alpha and SLC7A11 by COMMD10 depletion, suggesting that COMMD10 inhibited SLC7A11 by binding to and suppressing HIF1 alpha nuclear translocation in liver cancers. It has been reported that intracellular copper could stabilize HIF1 alpha protein, and a microarray database indicated that intracellular copper was positively correlated with HIF1 alpha in hepatocyte cells. Thus, we hypothesize that COMMD10 might modulate HIF1 alpha protein stability via regulating copper. HIF1 alpha expression significantly increased, and the half-time of HIF1 alpha was significantly prolonged in COMMD10 depleted cells under lomoxia and hypoxia condition, while TPA could reverse this effect. Furthermore, COMMD10 depletion could inhibit the ubiquitination and degradation of HIF1 alpha proteins. Also, this effect could be reversed by TPA. The above results indicated that COMMD10 depletion stabilized HIF1 alpha protein by augmenting intracellular copper. The effect of COMMD10 and HIF1 alpha on irradiation-induced ferroptosis was further confirmed in vitro and in vivo. COMMD10 overexpression obviously elevated the level of lipid prostatecancer and MDA, while HIF1 alpha could reverse this effect in irradiation-exposed liver cancer cells. Furthermore, tumors derived from COMMD10 depleted cells were larger and had lower lipid peroxidation than control group when irradiated. Conversely, YC1 could reverse the tumor-promoting effects. Constantly, COMMD10 depleted group showed fewer cell death area and higher ratio of Ki67 positive cells than control group, while YC1 could reverse this effect. Above all, COMMD10 inhibited HIF1 alpha stability by decreasing intracellular copper to enhance ferroptosis in liver cancer. Iron can trigger ferroptosis by inducing phaeton reaction. The results showed that the level of lipid peroxidation and MDA was significantly increased in irradiation-exposed group, but in contrast, iron chelator could decrease the level of lipid peroxidation and MDA compared to that with iron whether irradiated or not. These results suggested that iron played a critical role in irradiation-reduced ferroptosis in liver cancer. Iron decreased in COMMD10-depleted cells and increased in COMMD10 overexpressed cells, indicating that COMMD10 could increase the level of iron. Cancerosmeme, which is called CP here, is an important regulator of iron metabolism. So here we speculate that COMMD10 might regulate CP expression to induce ferroptosis in liver cancers. Our results showed that COMMD10 negatively regulates the level of CP expression, and TEPA could restore CP expression caused by COMMD10 depletion. Using COMMD10 and CP co-expression cells, we validated that the co-expression could restore the increase of iron, lipid peroxidation, and MDA caused by COMMD10 overexpression. These results indicated that COMMD10 suppressed the CP expression by decreasing intracellular copper to enhance ferroptosis. HIF-1 alpha has been reported to promote CP expression. To clarify their relationship, we analyzed the binding site of HIF-1 alpha and the CP promoter region through the JASPER website. The result showed that HIF-1 alpha bind to the CP promoter at HIF-1 alpha overexpression increased the mRNA and protein levels of CP in cells, which suggested that HIF-1 alpha promoted CP transcription and translation. Moreover, YC1 could recover the increased CP expression induced by COMMD10 depletion, suggesting that COMMD10 depletion promoted CP expression by enhancing HIF-1 alpha in liver cancer cells to irradiation. And I also validated CP could promote HIF-1 alpha expression by inhibiting iron. Here we found that CP overexpression increased the HIF-1 alpha expression, and COMMD10 and CP co-expression could reverse the decreased HIF-1 alpha expression induced by COMMD10 overexpression when irradiated, indicating that COMMD10 inhibited HIF-1 alpha expression by inhibiting CP expression. Overall, COMMD10 increased ferroptosis at radiosensitivity via inhibition of HIF-1 alpha and CP loop in liver cancer. The relationship among COMMD10, HIF-1 alpha, and ferroptosis was further explored in clinical specimens, which indicate they might be used as predictors for the radiosensitivity in liver cancer. To further explore the clinical application of TEPA and IC1, we treated irradiation-resistant HEP2 cells with increasing dose of TEPA and YC1. We found that YC1 and TEPA significantly reduced the proliferation of irradiation-resistant cells, and the same results were found in radiation-resistant mice model. These results suggested that copper channel TEPA and HIF-1 alpha inhibitor YC1 were two potential radiotherapy sensitizers for the radio-resistant cancer patient. Taken together, the whole story, no expression of COMMD10 induced intracellular copper accumulation. HIF-1 alpha upregulation and nuclear translocation by inhibiting ubiquitination and degradation of HIF-1 alpha, thereby upregulating downstream CP and SLC7A11 target gene, which inhibited ferroptosis. On the other hand, high concentration of intracellular copper upregulated CP expression to reduce intracellular iron concentration, which inhibited ferroptosis too. Upregulation of CP could also enhance HIF-1 alpha expression, forming HIF-1 alpha and CP positive feedback regulation loop and promoting radioresistance. This work provides new targets and treatment strategy for radioresistance in liver cancer. That's all. Thank you and welcome to Guangzhou.